Hi, my name is Abhi Karri, and my project is Col-IV, an indicator of vascular formation and pericyte recruitment. So before I get into the details of my project, I want to answer one question, which is, what are strokes and how do they impact the cellular environment? Stroke is a condition where brain tissue is deprived of essential oxygen, causing cells to enter a state known as hypoxia. Hypoxic cells drive vascular changes by proliferating rapidly, and ultimately they destabilize the extracellular matrix. No studies have really quantified the impact hypoxia has on ECM or the impact ECM changes have on the other cellular components. And so the focus of this study was to quantify the hypoxia's impact on the extracellular matrix, otherwise known as the ECM. Specifically, again, the main focus of the study was on collagen and pericytes. Collagen is a structural protein that plays key roles like supporting tissue growth and contributing to vessel stability, and is the most abundant protein in the ECM. Pericytes are specialized cells that play key roles in microvessel homeostasis and they contribute to the construction of the ECM, which is why they were also considered in this study. So moving on to how the experiment was set up. The experimental design was, in terms of the experimental design, it was hypothesized that if the cells are put into hypoxic environments, they'll upregulate protein expression in order to attempt to fix damage. But ultimately, cells won't be able to keep up this upregulation, and thus a drop in protein expression will be observed. The independent variable for this project was the hypoxic duration, which is at the 12-hour period and 24-hour period. Dependent variable was collagen density, pericyte presence, and gene expression levels. And finally, the control was double reporter embryonic stem cells that were cultured under regular conditions with no hypoxia. In terms of the methods, again, cells were cultured basically at the various levels required. Then I ran immunocytochemistry and qPCR analysis on the cells, conducted confocal microscopy and created an image stack, and finally I created a novel computational image analysis model for the image stack and recorded my findings. So after doing all that work, what was discovered? This slide shows an example set of images for running the confocal microscopy. The set of images on the left are confocal images for collagen-III, and the set of images on the right are for collagen-IV. Some general trends that were observed were that collagen-III volume decreased as hypoxia duration increased, which was seen in the left image set for the first column. However, something interesting was that DS-red signaling, which is the column to the right of that, increased in cells with 12 hours of hypoxia, indicating that more pericyte recruitment occurred, but at 24 hours, there was no signaling, which raises the question whether pericytes were actually able to differentiate under hypoxia or not. The image set to the right shows similar trends in terms of collagen-IV. However, the one difference is that for collagen-IV volume, which is seen on the first column on the left for the collagen-IV set, at the 12-hour period, there is more density, there's more volume there, but at 24, that volume decreases and sort of starts to normalize. And so I quantified all this into my graphs, and here's my collagen-III graph. I ran a two-tailed t-test for significance amongst all my categories and graphs, and I found a statistically significant difference between the control for the collagen-III and the 24-hour period in the collagen-III. From this data, it suggested that cells are unable to continue production of collagen-III in response to their hypoxic environment, and thus we see that decrease over time. Moving on to my collagen-IV graph, I found that there existed, again, another statistically significant difference between the control and the 12-hour period. From this data, it suggested that when endothelial cells are first afflicted with hypoxic conditions, their choice ECM to repair themselves with is collagen-IV. However, as hypoxia continues, cells are unable to sustain such levels of collagen-IV production, and so we see that trend. After observing these trends, I really wanted to understand what was happening on the genetic level, and so I ran qPCR analysis on target genes in hypoxic environments, and I found increases in Col-III-A1, Col-IV-A1, and Col-IV-A2, which appeared to be driven by endothelial cells as they responded to the damage caused by hypoxia. On top of that, I found PDGFR beta, which is also called platelet-derived growth factor beta, transcript increased upon hypoxic induction, suggesting that hypoxia results in decreases in pericyte recruitment at the 24-hour period, but not at the 12-hour period. Finally, moving on to the conclusion. To tie everything back to the beginning, my hypothesis was accepted by the collagen-IV data, but not by the collagen-III data. In terms of major findings, though, the data suggests that collagen-IV, again, is the cell's first choice of ECM to repair the cellular environment when under hypoxic conditions, which was supported by a Pearson correlation between pericyte recruitment and collagen-IV. Again, the reason for this is that collagen-IV could be a potential recruiter for pericytes as well as a potential solution to vessel damage, and that's why cells recruited to do a sort of two birds with one stone type of thing. Another finding was that hypoxic environments may impair cells' ability to translate mRNA into necessary proteins, as was seen from the qPCR data, and could be another avenue of further research. In terms of future studies, the study revealed a significant potential in developing novel, next-generation therapeutical drugs to address the decrease in collagen-III. Hypoxia isn't its own unique condition, but rather can be found in a variety of vascular conditions ranging from cancer to diabetes. And so by addressing this broad condition, we make headway in changing the field of medicine and research from simply being reactive to a condition to being proactive and preventing the conditions from returning or even starting.